Wangisa, Dunuwille. Wangisa got a Bachelor's of Veterinary Science degree in 2010 from the University of Cardinia in Sri Lanka. After graduation, he practiced as a veterinarian for 2 years. And then in 2012, he enrolled in a master's degree program in experimental biotechnology. He completed that in 2014 and then came to our program, joining Dr. Balasuria's lab and studying in the area of biology. Wangisa.
So I'm actually going to talk to you about viruses and abortions. So my research involves two viruses, equine herpes virus 1 and equine herpes virus 4. So as their names suggest, these two viruses infect horses. But that's not all. These two viruses are pretty similar to one another. In fact, for a long time, these two viruses were considered as two subtypes of the same virus. However, their pathogenesis is completely different. Equine herpes virus 1, more often than not, would cause abortions in pregnant mares. And also, occasionally, they would cause neurological diseases. In contrast, EHV4 does not cause neurological diseases and very rarely causes abortions. So, in the equine industry, EHV1 outbreaks have been considered, or it's more detrimental than EHV4 outbreaks. So, why is this? Well, it's probably not a single reason thing, but one of the most important reasons behind the difference in the pathogenesis of these two viruses is their ability to cause inflammation in the small blood vessels of the pregnant uterus. Now, inflammation is the normal response of living tissue towards infection or injury. And it seems like EHV1 strains are much more capable of inducing inflammation than EHV4. So, why is this? Why is EHV1 much better at causing inflammation than EHV4? This is the question that I am trying to answer with my research. So, the approach that we have taken so far is to assess the differential gene expression of cell surface inflammatory markers, pro-inflammatory cytokines, and chemokines induced by these two viruses when using in vitro cell culture models. And so this would give us an idea about what genes these two viruses might be upregulating or downregulating during an infection. And from the results that we have obtained so far, I can say that the gene expression profile of endothelial cells infected with these viruses have significant differences. And we hope that with further research, we would be able to further elucidate the reasons behind the differences in the inflammatory process induced by these two viruses, which might help in developing much better treatment and management conditions during disease outbreaks. Thank you.